He started in terms of human genetic research at the University of Tübingen and also Heidelberg. He's been working with embryonic and fetal cell extract therapy. The nice thing about Dr. Friedrichson is that he has both a PhD and MD. So he not only does the research part, but he also works on the practical side. Again, I'm not going to take much time from Dr. Friedrichson, but he's such a hard worker, works long hours, and I hope that one day you're going to be able to get your books done so that you can put down all the experience you have with the 10,000 cases of research and practicum that you have. And least of all, people don't know that Dr. Friedrichson is a really humanitarian. He volunteers his time on a regular basis in Africa. So again, welcome Dr. Friedrichson to Bangkok. Thank you.
Excuse me. Good morning, ladies and gentlemen. Thank you for the nice introduction, David. I'm talking about the cell therapy, the past and future of anti-aging medicine. The treatment method for the future was already mentioned in the papyri of the old Egyptians, and about 2,000 years ago in the writings of Paracelsus, a Greek philosopher and a scientist, when he proposed the principle of similia similibus curentur. Embryonal organ cells and their components are being used to revitalize and regenerate damaged, used, and aged organs or organ systems. We are passing through the evolution in fast motion during our embryonal and fetal development. That means that a fertilized egg on its way to being a baby passes in 9 months through the whole evolution from a one cell to the biosystem Homo sapiens. We are passing through the state from an undifferentiated protozoan to a fish, to an amphibian, to a mammalian, and finally to a full differentiated human being. The genes, and that answers also a question from this morning, in our embryonal and early fetal stage are not different in their sequence compared to our mammalians, and this is very important for the cell therapy. We could already show in the 1970s that the protein sequence of enzymes used in the early stage are identical with other mammalians. So that showed clearly that a genetic variability is only allowed in the evolution where life, that means survival of an organism, is not in danger. So that shows that the genes are in the early stages are the same used in mammalians as in humans. And as Dr. Ali Mohammed said, the cell therapy, which is using from this kind of biological state, proteins or cells have the same active genes, repair genes, what I come later to, as the humans. We have therefore genes activated in this early phase of our development which carry information for a growing organism, in our case, regenerating organism, which are the same in all mammalians, and that's a take-home message. Furthermore, all organisms which are more advanced in the evolution have so-called repair genes in their genome. Those are present in maximum amount and maximum activity in the embryonal and fetal status. The more mature, that means older or damaged an organism is, the lesser amount of numbers and activity of those genes we have. That means simply the older we get, the sicker we get. The clinical cell therapy has an immense potential in treating injured organs, help to regenerate tissues and organs, as humans as a whole biological system. As a consequence of these findings, those therapies are not only able to heal, but really to regenerate, as we saw this morning at Ms. Dr. Mohammed, that we can stop, at least slowing down even the biological clock. The united cell structures, cell components we use in the cell therapy are embryonal, early fetal, and seldom early juvenile. They are kept in physiological media and furthermore administered or implanted, as we say in cell therapy. And again, to repeat Dr. Mohammed, we have here no antigenicity but organ specificity. So that's why we can achieve, if we take it from the sheep, for example, good tolerability and good organ activation. The cell therapeutica we use have an essential advantage compared to normal transplantation of adult tissue. Since our cells, components, growth factors have none or close to none antigenicity, the growing organism has to be tolerated from the mother body, as we know. Under physiological conditions, there is no anti-gene production during pregnancy. Over the last 35 years of our own research and therapeutic experience, we have never seen a rejection in thousands of implantations. The transplantation of adult tissue for heart, lung, kidney, pancreas, as you know, show that without suppressing the host's immune system would result in life-threatening organ rejection. The activity, the reactivity and ability to repair from embryonal and early fetal tissue is uncomparable and extremely efficient to adult tissue. The activity and biological repair potency of cells and their components, including growth factors, of such an early biological stage have a tremendous advantage, so that cell therapy are not only leading the way in treating diseases which we could not handle before, but in our opinion will be the gold standard in the anti-aging medicine of the future. Somatic cell lines are highly differentiated, as we heard this morning. That's the positive part, but the capacity to reduplicate is very limited. That's the negative part. The activity of the genes, cell organelles, cell components, and growth factors are used in the cell therapy to regulate and stimulate metabolism to support aging, aged, and defect tissues. It could be shown in uncountable research papers that embryonal and early fetal cells are packed with huge amounts of highly active genes, DNA, RNA, growth factors, enzymes, and other cell organelles, mitochondria. This biological equipment starts after implantation in the host organism, repair procedures, and optimizes regenerative and revitalizing processes. To implant a large amount of connected cell, like a lump, cell or cell formations was not very effective and of great value. It could be shown, however, that when we implant xenograft cell suspensions, cell organelles, cell parts, growth factors, specific proteins, we had never had any problems with absorption and acceptance from the host organism. It was already shown that in the last century by radioactive marked cells and cell parts that there was fast uptake and incorporation in the host organism. But not only that, it was found that there was an exact organ specificity. Liver cells or their parts were transported to liver, heart cells were transported to heart, so kidney to kidney, brain to brain. Furthermore, it could be shown that the implanted cells or cell parts did not dock to the effector organ as a whole lump, but the cell parts were fast incorporated in the existing tissue, and the whole cells were as single cells attached to the organ. The last 10 years, lots of publications showed by each cell division telomeres shortening occur to each chromosome. Inborn genetic error increase, numbers of active repair genes decrease. The degeneration is an ongoing process. As we say, life ends always deadly. During the pre-implantation period, the cells or cell parts pass through a specific preparation that it makes it extremely absorbable. Here we see a picture of undifferentiated human embryonic stem cells. And aged and damaged tissue can be altered, activated, and repaired due to mentioned organ specificity. Since the first publication of the and description of the so-called cell crosstalk, we have an exceptional and marvelous biological proof of what we postulated many years ago, that the information which we give to a host organism through the cell therapy is transmitted not only to a few cells of an organ, but to the whole biosystem human. Those cells are being activated so that it is not only the action of transplanting a few millions of embryonic or fetal cells, but through the biological principle, is it possible to reactivate the existing tissue and have a much higher effectivity and improvement of single organ systems and the whole body itself. So you see those cells are gluing together after a while and make channels and give information and incorporate more proteins, repair genes, and so on. Just to show you in this picture the improvement of the kidney function through cell therapy by means of creatinine and glomerular filtration rate. It was a small cohort of 97 patients, and the decrease of the creatinine was more than 90% to normal levels within 6 months. And the glomerular filtration rate stepped up to normal in the same rate. The next, even more impressive study was done in 41 patients aged 64 to 87. It showed by neuropath procedure done after the beginning, after 4 weeks, and after 4 months, the improvement of oxygen uptake after cell therapy. And we could show that even after 4 weeks, already 75% was an improvement in oxygen uptake in the brain. And after 4 months, it was 182%, highly significant. It could be shown that the existing research results of the last 50 years that embryonal and fetal tissue have a massive stimulation effect of adult tissue. In our lecture this afternoon, we show the effectiveness of cell therapy in Parkinson's and Alzheimer's diseases, just to mention a few. Used cells or cell parts are passed on, which are not used are passed on to the apoptosis, the programmed cell death, without damaging the host organism. But not only that, the so-called spare parts can be used by the body and by the host's body. It could be shown that after the application of xenotransplants, that means not from humans, the cells or cell parts arrived at the destination organ after about 24 hours or less. Following this, the activation process needs understandably between 3 and 7 days for converting the cell metabolism in the direction of repair and regeneration. In mesenchymal tissue, kidney, brain, nerves, is the minimal reaction time 3 days to 3 weeks. In endocrine and placental tissue, we see sometimes reaction within hours to very few days. One of the most and well-known cases was stated this morning in the medical history is a lady from Switzerland which had by accident her complete thyroid and parathyroid removed. Without help, she would have died within a few days. Only one of the founding fathers of the cell therapy, Paul Nihans, treated her with cell extracts, and she survived and lived healthy for more than 30 years on and died in her late 90s. Now to anti-aging medicine. We could show in 2 age groups, 76 patients, age group 42 to 52, and the other one was 95 patients with 63 to 75 years. Done the measurement of skin elasticity with a cutometer. As a baseline, we took the elasticity of patients between 20 and 22, and we could show that we had an improvement in the younger group from 43% to 78% compared to the 20 to 22-year-old cohort. And the older patients went from 38 to 68%, which is also highly significant. In our opinion, the cell therapy is the most biological treatment one can get. Why? We always treat causes, not symptoms. Physiological and psychological exhaustion, early onset aging, general lack of vitality, immune deficiency, malfunction of endocrine glands are prime examples for cellular biological anti-aging medicine. Tens of thousands, no, hundreds of thousands of people improved through this therapy. Celebrities, as we saw, and ordinary people over the last 100 years. So what we can definitely state here is with cell therapy, we give first the years more life, and second, the life more years. Thank you.
Well, thank you very much.
Please contact the German Society for Thymus Therapy for further information.